Welcome to Biochemistry 601, Lecture 20. Today we're going to talk about ATP binding motifs. This is a wonderful opportunity to learn something about a large class of proteins. About 10% of proteins in E. coli are ATP dependent. About 9% of annotated proteins in the human genome are ATP dependent. So this is a great opportunity to learn the common features of a very large number of proteins. These enzymes catalyze phosphoryl transfer reactions. And in many cases, there's a direct coupling between the transfer of the phosphoryl group to the chemical reaction so that it favorably moves a reaction that would otherwise be thermodynamically unfavorable. And in these cases, there is generally a direct chemical involvement of the phosphoryl group in the reaction chemistry. There are two types of phosphoryl transfer reactions: an alcohol amide or water to the gamma phosphate, or a transfer of an organic moiety, an alcohol moiety, to the alpha phosphate to eliminate pyrophosphate. And in this case, it will form a covalent AMP dependent intermediate. And in this case, this can really force thermodynamically unfavorable reactions towards the product because the pyrophosphate is subsequently hydrolyzed and releases a significant amount of free energy. Now, our understanding of these enzymes has been helped enormously through nucleotide analogs, and these are really versatile tools for understanding protein structure and function. They get great for structural studies. They're also terrific for kinetic studies because they allow us to trap proteins at important catalytic states. But it's important to remember that they're analogs. They're not ATP, so that each of them has a drawback. There are two general classes of nucleotide analog. There are the non-hydrolyzable analogs, where an atom in the nucleotide is replaced by a nitrogen, carbon, or sulfur. And then there are the vanadate or metallofluoride complexes that form spontaneously in the active site to mimic either the ground state or the transition state. The common non-hydrolyzable analogs that you will encounter in your studies are ATP gamma S, AMP PMP, and AMP PCP. These are the acronyms for the replacement of either a terminal oxygen with a sulfur, a bridging oxygen with a nitrogen or a carbon, NH group or CH2 group. The best of these analogs is ATP gamma S. The downside to ATP gamma S is that it's slowly hydrolyzed, both in solution and on the enzyme, so that you have to be very careful to make sure that your ATP gamma S solution is not contaminated with ATP. But even so, it really is a terrific inhibitor. It binds very tightly to enzymes, most mimicking very, very closely ATP. The two that are very commonly used, probably the most common, is AMP PMP. You have the replacement of an oxygen with an NH group, and in this case, you can expect to lose one hydrogen bonding interaction. And as a consequence, typically AMP PMP does not bind as tightly to the enzyme as ATP. Of course, with AMP PCP, you replace the bridging oxygen with a methylene group, and this results in the loss of two hydrogen bonding interactions, and frequently it binds very weakly. So you don't see AMP PCP used all that often. If we replace the bridging oxygen between the alpha and the beta phosphate, that gives analogs that are useful for studying enzymes that release pyrophosphate and generate an AMP acyl intermediate. So in particular, you will see the AMP PCP used frequently because this is commercially available. This is generally not quite as often as available. The next class I'd like to talk about, and these are now very commonly used, are the metallofluoride type analogs. And these, as I mentioned, form spontaneously in the active site. Typically, they're prepared by combining in solution your enzyme with magnesium ATP, sodium fluoride, and a metal salt. It's important to remember that in ATP-dependent enzymes, they're almost always magnesium dependent. So the dissociation constant for magnesium ATP is about 2 millimolar in solution, so you must expect that your ATP comes in with a metal ion. The most common complexes that are seen in the literature are beryllium, aluminum or gadolinium, and sometimes scandium and magnesium, and we'll talk a little bit about this later in the talk. And they've been really very helpful for P-loop enzymes and protein kinases. They've not worked for ATP grasp enzymes, as we mentioned. I show here the structures of the beryllium fluoride, aluminum fluoride, magnesium trifluoride. And you'll notice that these have different configurations of atoms: square planar, square, trigonal bipyramidal, and tetrahedral arrangement around the beryllium fluoride. And as a consequence, we know that for beryllium fluoride, which I've shown X here because sometimes the fluoride is replaced by a hydroxyl ion, which means you have to be very careful about interpreting the interactions of those fluorines or hydroxyls in the active site when you determine their structure. Typically, because it forms a tetrahedral coordination complex, this mimics ATP in the ground state. Conversely, aluminum fluoride forms a square planar arrangement, and occasionally a trigonal, which is almost certainly magnesium trifluoride, as we now know. And these mimic the transition state for phosphoryl transfer that we'll get to in just a moment. As the phosphorus is being transferred from the beta oxygen to the receiving oxygen or nitrogen, the phosphorus will be transferred. It will go through a planar transition state, and these mimic then the transition state. The last class of inhibitor that you will see used frequently is vanadate. If you take sodium orthovanadate in solution, mix it with your ATP, ADP at the correct pH, around pH 7, you'll spontaneously form in the active site a trigonal bipyramidal complex between the vanadium ion, 3 oxygens on the vanadium, a terminal water, and an oxygen from the beta phosphate. Interestingly, this was discovered as an inhibitor, as a contaminant in one of the early isolated ATP materials that you could buy from Sigma. It's a terrific inhibitor, and it mimics the transition state. It's important to note it mimics the transition state. It is not the transition state, because the transition state is something that's fleetingly present. So it allows us to see the conformation perhaps of the protein close to its transition state, but it is not the transition state itself. That's a very important point to remember. And the same is true for all of these analogs. You have to think about, they're not ATP, they bind tightly, but how do they differ from what we would expect ATP to be? So you have to be careful about vanadate, because in the case of the vanadate complex, these oxygen-vanadium distances are quite plastic, so they may not mimic exactly what you would expect to see for ATP. So the analogs are terrific. You just have to be careful about interpreting them and not interpret them too far. So for the rest of the lecture, I'd like to talk about ATP binding motifs and what we can learn from them we can apply to all ATP dependent proteins. Now, there are many folds that coordinate ATP. You will find there are a few that are particularly abundant, and we'll talk about those. So we'll talk about the P-loop motif, the ATP grasp, and the motif that's seen in serine-threonine kinase domains. So the goal here is to understand why they have evolved, how they've evolved to coordinate ATP, remembering that they're all recognizing the same ligand, magnesium ATP. So what features should we look for in an ATP dependent enzyme? Well, coordination of the base. You want it to be specific for adenine, not guanine, or any other pyrimidine base. It should be specific for it, because the last thing you'd want to do is utilize one of the less common nucleotides in a metabolic reaction. You'd want to look for something that coordinates the triphosphate moiety. Carries 4 negative charges, generally in solution at pH 7. And you would expect to see a divalent metal binding site, remembering that your enzyme is going to probably accept magnesium ATP as the substrate. And then you would look for catalytic groups to facilitate the phosphoryl transfer. So the first group that we'll look at is an abundant set, and these are the P-loop proteins. So what I want you to get from here is the big picture of how this class of protein binds ATP. Now the P-loop itself was a sequence motif that was recognized first in F1ATPase, and it's also found in adenylate kinases, myosins, kinesins, G proteins, transporters. So it's a really abundant dynein. It's a really common motif. There's a consensus sequence, and it's glycine, followed by 4 residues of variable nature, glycine, lysine, threonine, serine. And it's sometimes called the Walker A motif because it was identified by John Walker, who was studying F1ATPase. In this case, the ribose oxygens, which I haven't spoken about, are often exposed to solvent. In this particular case, this is not true for most ATP binding motifs. Remember, it's magnesium ATP. So the conserved residues have a purpose. You don't have to remember this. You have to remember the general features, but you don't have to remember the motif itself. I want you to understand the roles of the residue and why they're appropriate. So if we look at the P-loop motif as observed in thymidylate kinase, what you see is that the motif itself starts at the end of a beta strand, goes through a loop, hence the name loop, and consists of the first half of a turn of alpha helix. And so typically this is one component of a larger sheet, and then there are a series of alpha helices that lie on top of that sheet. In this case, the enzyme catalyzes ATP, thymidine phosphate to make ADP and thymidine 5-phosphate, and serves to activate the thymidine 5-phosphate. This is a ribbon representation. We'll talk about the Walker B motif next. Now, there's an additional sequence found in many P-loop dependent, P-loop containing proteins, and this is known as the Walker B motif. And it is consists of a consensus aspartate, something, something, glycine. And it plays an important role in metal coordination. That's the aspartate, the negative charge. And it's often involved with conformational switching in those cases where there's a conformational change that happens in the active site. And we'll talk a little bit about that in the next lecture when we talk about motor proteins. Now, as you would expect, hydrogen bonding is really important for coordinating magnesium ATP in the active site. And this shows the interaction. It doesn't include all of the hydrogen bonding interactions, but to a first approximation, every oxygen of the phosphate participates in a hydrogen bonding interaction, both with the protein and with the magnesium ion. Magnesium ion also is coordinated by the ATP, protein ligands, and by water molecules. The magnesium ion is always octahedral. So when you look at the structure, you should expect there's something wrong if the coordination is not octahedral, because that's found in all P-loop dependent enzymes. So what are the roles of the amino acids in the consensus sequence? This is something that sort of general take-home message. And this shows an alignment of a series of proteins, and you don't have to remember the proteins itself. It's the big picture that counts. You'll notice there's a completely conserved glycine. There are additional glycines, although not in a particular position. And then there's a conserved glycine, and then there's the lysine and threonine. The glycines play a particularly important role here because they allow that polypeptide chain to adopt conformational angles that are not available to the other amino acids. And what that allows the backbone to do is to wrap around so that it can place the amide hydrogens pointed towards the oxygens of the phosphoryl oxygens. This is important because it presents a fairly fixed framework that puts the hydrogen in just the right orientation, which increases the binding affinity. So this brings us to an important point. Although they contain glycines, the loop itself is not flexible. It is held in place by secondary interactions with other components of the protein, so it's not flexible. There are some enzymes where that conformation of the P-loop changes during the catalytic cycle to reduce the affinity. But even in those cases, the P-loop is still well organized. So the residues that face away from the loop play an important role in stabilizing the loop. Now there are two evolutionary groups of P-loop proteins, probably more than that, but there are two that are entirely obvious. And one of those, these are separated into groups by whether or not there's an additional strand that lies between the P-loop motif and the Walker B motif. So they're either, the Walker B motif is either adjacent or there's an additional strand between the two. And these are listed here. Again, you don't need to remember this. It's what it's leading up to is how these enzymes have evolved to recognize ATP. So there's an extra strand in the P-loop kinases and motor proteins that we'll talk. In this case, it is a GTP protein. Dependent proteins, the G proteins, have the P-loop in them, so they utilize GTP. And in this case, of course, they're functioning as switches. So let's look at the structures of a representative group that, where they're adjacent or where there is an additional strand that lies between the P-loop motif and the Walker B motif. What you will notice is that the conformation of these three, of GTP, ADP, and ADP in this case, are all very similar in nature. So what about the Walker B motif? I mentioned that it coordinates, provides one coordination point for the magnesium ion. So if we superimpose the P-loops from thymidine kinase and RAD-A, which is a REC-A, so the thymidine kinase does not have an extra strand. The REC-A does have an extra strand between the Walker B and the Walker A motif. What you'll notice is that the coordination of the ADP, ignoring what happens at the adenine, that's not conserved. But if you look at the overall conformation of the triphosphate-like moiety in the active site and superimpose the active sites based on the orientation of the triphosphate, what you will see is that the P-loops are essentially identical. And remarkably, the position of the aspartate group on the Walker B motif is in the same position to coordinate magnesium, regardless of whether or not there is an additional strand there. So what it tells you is that the evolution of this class of binding proteins has been driven by the structure and chemistry of ATP. So they've evolved to recognize the unique features of this ligand. So I'd like to spend a moment or two to talk about the chemistry of phosphoryl transfer. So phosphoryl transfer consists of the attack of an OH, an oxygen, the lone pairs of an oxygen on the gamma phosphate and its nucleophilic attack, and this is drawn as it should be drawn, rather than as is typically drawn with a double bond. All of the bonds to the ATP have the same length. They're not, as Hazel has mentioned earlier in the lecture, so this is a better representation. As this oxygen attacks the alcohol moiety, be it water or an organic alcohol, you're going to have to remove the proton that's present here as it attacks. Either you're going to remove the protein first, and it could be said to be a catalytic base, or the proton's going to have to be transferred as it attacks the phosphate. So when you look in these active sites, you'll ask the question, what's the base? What's the catalytic base? And there are some enzymes, and we'll talk about this with the motor proteins. It appears that the ultimate base is actually the gamma phosphate itself. You can't see in this particular class, the myosin, for example, you can't find a catalytic base. So the nature of the transition state. All right. So the gamma phosphoryl moiety is transferred from ATP to its receiving nucleophile, be it water or an alcohol. We understand from physical chemistry that this can either go through a dissociative type transition state where one has weak interactions between the oxygen and the phosphate. And in the middle is a moiety that is metaphosphate-like in species. Metaphosphate has been observed in, and that's PO3-. Metaphosphate has been observed in organic reactions, and it's a fleeting species in water. The alternative is that one has a more associative interaction where one has substantial bond buildup between the attacking oxygen and the departing oxygen, the leaving group, so that there is much more bonding character associated with it. There's an excellent review that was published in 2011 that goes over this particular subject, and I recommend it to you if you're interested to probe deeper. What we understand is that for enzymes, the transition state for the phosphoryl transfer is metaphosphate-like. It's not metaphosphate, it's metaphosphate-like. In other words, the transition state occurs when the 3 oxygen and the phosphoryl group, phosphorus atom, are in a plane. And that, of course, explains why vanadate and magnesium trifluoride are such excellent inhibitors. Now, the next class of proteins that I'd like to discuss are the ATP GRASP proteins. And these are completely different motifs, completely different protein folds. And they catalyze a different type of chemical reaction. Here, it's not the reactions themselves, except for the general trend, that's important, the general class of reaction. What I want you to think about is how is ATP coordinated? It's coordinated differently. But what is it doing? What is it recognizing? And how is it accomplishing the task of coordinating ATP? So the biochemical properties of ATP GRASP are that they catalyze the condensation between a carboxylate moiety and an amine-like moiety. And the role of the ATP is to activate the acyl group of the group that is going to be transferred, the carboxylate group. This is a very unstable moiety, but it's real. This is a real component. And then it will react readily with the nitrogen to form an activated, an acyl conjugate between these two. And the presence of a phosphoryl moiety on the acyl group serves to activate this process. Otherwise, it would not be driven to completion. So the acyl phosphate is an important intermediate, and it's found throughout life, prokaryotes, eukaryotes. In this case, they can all be described, this class of enzymes, the ATP GRASP, as having 3 domains. And the ATP binding pocket, as opposed to the P loop, is wedged between the B and the C domains. So if we look at the types of reactions then that are catalyzed by this group of enzymes, it's really diverse. They can transfer bicarbonate, biotin carboxylase. In the bacterial peptidoglycan biosynthesis, dealanine ligase, so the substrate is alanine. You can see what you're accomplishing, what you're activating is highly diverse, but there are common features in their structures. If this class of enzymes was not recognized for a long time, because the sequence signature, as opposed to the P loop, which is entirely obvious, you can spot that by eye, the sequence signature is distributed throughout the protein so that it's very difficult to, at first sight, to find the similarity. But this shows a significant number of sequences that were aligned, and you can see that they're distributed, and you can see, yes, there are conserved glycines, but it's not as obvious as a nice short P-loop sequence. It actually only became obvious that they were similar when the structures of representative members of this class of protein were determined, and then it became obvious which residues and why were important for this class of enzymes to function. And again, the conserved glycines play a role in the phosphate binding. again because they serve to place, allow conformations of the backbone that would not be allowed with other amino acids, such that the amide hydrogen can be directed towards an oxygen of the phosphoryl, the triphosphate moiety. So as I mentioned, the active site is distributed. Apart from the fact that it's distributed, you don't have to remember this motif. I typically say that if you have a slide, you should talk about everything that's on the slide. Well, clearly, you can't even read this, but it's the general trend that the sequence motifs that are important for binding are distributed throughout the protein. So we'll take one example. We'll look at one example in a little more detail. And this is the purity-encoded glycinamide ribonucleotide transferase. And it's important for purine biosynthesis. Purine biosynthetic pathways complex. It consists of 10 to 11 enzymatic steps. But one of the important steps is the activation of this glycinamide ribonucleotide. And what we're going to do then is we're going to build up the purine ring. So in other words, we're going to have to add carbon atoms, and that's a tricky thing to do. And the first step is to activate bicarbonate to generate, in this process, activate bicarbonate, and then transfer this COH moiety, the formyl moiety, to the substrate. This is obviously an unfavorable reaction. Once you've accomplished this, you can see that I'm starting to build up the carbon atoms necessary to form the 5-membered ring of the purine. And this catalytic process is a matter of adding on methylene groups one at a time to be able to facilitate the synthesis of purines. As I mentioned, the pure T is an ATP grasp enzyme, uses ATP. The alternative uses formyl tetrahydrofolate, which takes you back to Hazel's lectures on coenzymes as a way of alternatively adding in the additional methylene group to accomplish the same overall goal. All right, let's take a look at the structure. How does ATP bind? Remember, the purpose of this lecture is to understand something about ATP binding. So in this case, the ATP binds between two domains, a central domain, and it lies as wedged between in a cleft. And there's a 4-stranded anti-parallel beta sheet that lies at the base. So in this case, the ATP binds in an extended conformation, and it's enveloped between these domains. It's completely enclosed by the active site cleft. As I mentioned at the beginning, what are we going to look for when we look at an active site? We're going to look for how it coordinates the triphosphate and facilitates the transfer of the phosphoryl moiety. But we also want to look in the active site for those parts that are responsible for discriminating between ATP and GTP. And if you look in the active site, you will see that there is an interaction that we'll mention again in the next slide that selects for ATP as opposed to GTP, ATP, GTP. GTP would not be recognized, would not bind stably in this. And then it binds to the active site, and in this case there are 2 magnesium ions. So the ATP is an extended conformation with the magnesium ion on either side of the triphosphate moiety. Coordinates to both the alpha and the gamma, beta and the gamma phosphate. So again, this brings across the point. If you look at this, it's utilizing binding determinants from throughout the protein. So this just shows it in a little bit more detail. And the important thing to focus again is that there is extensive interactions, extensive hydrogen bondings between all of the oxygens of the ATP. And the magnesium ions, or they don't look it here with this particular representation, these are octahedral in coordination. And again, the discrimination between ATP and GTP. In this case, as I mentioned, this is an example where the ribose moiety is buried. And of course, if you bury a ribose, then you're going to have to form hydrogen bonds. And in this case, there are hydrogen bonds that form to a protein ligand. protein amino acid side chain, glutamate, and also a water molecule. So the ribose oxygens are not accessible to the exterior bulk solvent, although there is one water molecule present. Now what's interesting is if you take a look at this motif that binds it from a series of a range of enzymes, what you notice is there is a loop connecting two strands that lies adjacent, parallel to the triphosphate moiety. And throughout ATP grasp enzymes, the coordination, the overall conformation of the residues that coordinate the ATP are remarkably similar. There are differences in the loop section that's to do with whatever else the enzyme is doing, but the moiety, the protein moiety that interacts with the ATP is conserved. And again, what this point is pointing out is that these enzymes have evolved to recognize a common ligand. So this overall conformation is driven by a preferred orientation, conformation of ATP. Now, in opposed to the P-loop proteins that generally don't undergo a large conformational change in their catalytic activity, in the ATP grasp enzymes, there tends to be a very large conformational change associated with the catalytic cycle. So if we look at biotin carboxylase, which is part of the acetyl-CoA carboxylase enzyme that activates biotin, so it serves to actually activate, using biotin, it activates bicarbonate. It's a way of adding an additional carbon atom. So if you look at the structure of the enzyme in the presence and absence of substrate, what you notice is that in the absence, the domains are fairly open, and the B domain is 70 degrees away from the active site. Upon coordination of the ATP in the active site, the B domain folds over the active site. This does a couple of things. First of all, it serves to exclude water from the active site. As I mentioned, these enzymes catalyze the activation of an acyl phosphate. Acyl phosphates are very unstable in solution, so a key component of these enzymes is that they have to exclude water from the active site to avoid futile hydrolysis of ATP. So yes, there are at least many contain a Rossmann fold. Beta domains, these are the strands that are common, and these are because they are recognizing a feature of ATP. And so that's the overall summary. So what do I want you to take from the ATP grasp? They're different, it's conserved, it's got two metal ions, it's recognizing a stable conformation of ATP, and it's binding, it's accommodating all of the features of the ATP that you can. You've got to take care of the negative charges on the phosphate. Those are the magnesiums, and typically you have lysines and arginines in the active site. You have to be able to, in this case, bind the ribose, and you have to be able to distinguish between ATP and GTP. Now, the next class of enzymes that I'd like to talk about are the serine, threonine, tyrosine kinase class of families. This is a very important class of enzymes in eukaryotes. It's a very large group, and it appears that they've all evolved from a common ancestor. So we're transferring a phosphoryl moiety from ATP to a hydroxyl side chain, hydroxyl group on a hydroxyl containing side chain. And they're important for switches, so they play an enormous role in signaling. Again, this is a switch. So the phosphorylation functions to change the properties of the protein as a consequence of having a phosphoryl moiety on them. They're as abundant in eukaryotes, and this shows a tree. When the first sequence of the human genome was determined, they could then identify the kinases that were present in the genome, and it's a very large group of enzymes, and that's why, in fact, 9% of the genome, the human genome, is ATP dependent because of the large number of kinases that we have. And they all appear to have evolved from a common ancestor. And what that means is they all have the same overall fold for binding and transferring the gamma phosphate to their substrate. Although, of course, what they recognize will vary enormously in how they are regulated. So the typical kinase domain, as shown in cyclic AMP-dependent protein kinase, so this is the domain that transfers the phosphoryl moiety to the substrate. It shows that this is a two-domain protein. ATP is binding in an extended conformation. So this is similar to the ATP grasp as opposed to, in the conformation of the ATP, as opposed to the P-loop protein. And in this case, the phosphoryl moiety is transferred to a 20 amino acid peptide that is found in a naturally occurring inhibitor peptide. So the protein in this case is phosphorylated, so we're looking at the product of the reaction. So on the basis of this and the sequence similarity, it's assumed that the common mechanism is utilized by all kinases for phosphoryl transfer. So if we look at the binding motif, and again, we're focusing on how does this enzyme recognize ATP? How does it accommodate the chemical features of ATP and its binding? Because they evolved to recognize ATP. Again, it binds in an extended conformation, and there are two metal ions. So this is more similar then to the ATP grasp. In both cases, the gamma phosphate faces into an opening that coordinates the phosphate acceptor. So again, this is reflecting the chemistry that is needed to transfer the phosphoryl moiety. One needs to place it into an opening where you can bring in the nucleophilic acceptor opposite the phosphoryl moiety so that you can transfer it. So the chemistry is driving the overall configuration of the active site. So if we take a close-up look at the ATP binding site, and we'll look at more detail in a few slides, what you will see that one of the important coordination components is a loop that lies on top of the, in this case on top, lies adjacent to the triphosphate moiety, and it provides hydrogen bond acceptors through the amide hydrogens to the oxygens on the gamma and beta phosphates. The two magnesium ions lie on the opposite side of this loop, and these are in the C-terminal domain. So this is in the N-terminal beta sheet domain. This is in the C-terminal domain that is mostly alpha helical in nature. It's a loop. So again, what is happening in this loop is that the glycines, yes, they're providing interactions, but they allow the loop to form a conformation that otherwise would not be available were they replaced by other amino acids because of the greater conformational freedom of the glycine moieties in the active site. So the take-home message here, two metal ions, close coordination. The glycine provides, the loop provides a mechanism to coordinate, to provide a good binding site for the gamma and beta phosphoryl oxygens that would otherwise not be available. And this loop in general is not that flexible. There's perhaps a little more flexibility than the ATP GRAS proteins, but most of the flexibility occurs between the two domains. You will see in the literature that sometimes this is called a P-loop, and it's not. It's a phosphate binding loop, yes, but a P-loop, that acronym should be reserved for the class of proteins that were first observed in F1 ATP. So you need to be careful because if you use the word P-loop, you're conveying a different meaning in this enzyme compared to the very large class of enzymes that we will talk about in motor proteins. Now there is a similarity between the ATP binding in ATP grasp and the protein kinases. In some ways, that was obvious from just a simple comparison. But if you line them up, what you will see is that the overall arrangement of the magnesium ions, the overall conformation of the ATP, in this case it's ATP, in this case it's AMP, PMP, positions of the magnesium ions, the overall orientation of adenine moiety are basically identical in both classes of enzymes. Yes, there's a change in the conformation. There's still a loop involved that provides coordination. But what it tells you is that these enzymes have evolved to recognize a common conformation, a stable conformation for ATP. So the evolution has been driven by the conformation of ATP first and foremost. I'd like to spend a couple of moments talking about the mechanism for phosphoryl transfer in the protein kinases. And some time ago, a structure was determined for a cyclic AMP-dependent kinase complexed with magnesium ADP, aluminum fluoride, and a substrate peptide. And this shows the figure from the original paper. This was published a long time ago, 2002. But it's a beautiful piece of work. And so they determined in the presence of aluminum, they said aluminum trifluoride. They added aluminum trichloride to a solution of sodium fluoride, which in solution would form an AlF4- ion. But they claimed that they saw aluminum trifluoride in the active site, magnesium ADP, and the substrate peptide. What we now know from studies 10 years later, that the aluminum fluoride was almost certainly magnesium trifluoride. Even so, regardless of the nature of the ion that is present, the exact nature, this mimics the structure of the protein close to the transition state. In other words, the aluminum fluoride, magnesium trifluoride is a mimic, and I stress the word mimic, for the metaphosphate-like moiety that would be present at the transition state. And you can see that it binds between the two motifs and lies in the cleft, and there's the trigonal bipyramidal moiety lying in the active site. And then there's the peptide to which the phosphoryl moiety would be transferred to a serine. And this shows the electron density that was observed. So it really is trigonal bipyramidal in nature, and it's coordinated between the ADP oxygen and the oxygen of the serine to which it would be transferred. And as I said, this is almost certainly magnesium trifluoride, so, but that wasn't known at the time that magnesium trifluoride could function as an inhibitor. So if we look at the coordination of the transition state analog, we can learn a lot by studying this carefully. It tells us a lot about how these enzymes transfer the phosphoryl moiety to their acceptor substrate. It also tells us a lot about regulation of these enzymes. Kinases are highly regulated, and they also don't spontaneously hydrolyze ATP. Their spontaneous hydrolyzation rate of ATP is exceedingly small. If you look at the active site, you will see the coordination for the magnesium ions, and you can see that they are octahedrally coordinated. The bond distances that you would expect to this moiety are the sorts of bond distances that you would expect to see for a magnesium-oxygen linkage. So they're around about 2 angstroms. It's about 2 angstroms to the magnesium. So these are mimicking the oxygens that you would expect, although they are not the oxygens of metaphosphate. And then you have a slightly longer bond distance between the central metal and the oxygen on the beta phosphate and on the phosphoryl, on the acceptor oxygen of the serine. So it's halfway in between. So it's mimicking what you would expect to see at the transition state. Now, if you look a little bit further away, apart from the fact that there's lots of coordination, if you look a little bit further away, you'll notice that the serine moiety is in close proximity to the aspartate group of 166. Now, this is almost certainly protonated, so this is a hydrogen bond. So this suggests that aspartate 166 functions as a catalytic base, and were this a normal ATP, you would expect that this would accept the leaving proton from the phosphoryl moiety. And then you'll notice there's a lysine, there are additional residues that serve to coordinate and stabilize this trigonal moiety in the active site. In other words, they're preferentially recognizing the transition state for phosphoryl transfer rather than the ground state. So, yes, and you'll notice this forms an important interaction. Right, so this brings us to the question of then, okay, this is the way it looks with ATP in the active site at the transition state for phosphoryl transfer. Why doesn't it hydrolyze ATP in the absence of this phosphoryl moiety, this oxygen on the substrate to which it would be transferred? And the answer to that can be seen by looking at the structure of ATP in the active site for this enzyme. And what you will notice is that the position of the hydrogen bonding is such that it's not possible for a water molecule to be coordinated readily to transfer directly down the beta oxygen, gamma phosphate, phosphorus oxygen vector through here. So that in order for a water molecule to attack, you would have to hold back the two protons, and the aspartate moiety would have to be in the correct orientation, which is not in this case. You would have to be able to accept a proton from the attacking water molecule. So the water molecules, the active site is not configured to allow a water molecule to be coordinated. It is the binding of the peptide in the active site, in other words, the auxiliary amino acids and their backbone interactions, that configure the active site for transfer of the phosphoryl oxygen. Now, last thing I'd like to mention is kinases as drug targets. As you are all aware, if you watch television anytime, you'll see lots and lots of advertisements for drugs that target kinases. Now, at first sight, you would not expect a kinase to be a good drug target. And the reason for that is they bind ATP, and 10% of enzymes bind ATP. So you would think that an analog that recognizes that's like ATP would be a lousy inhibitor, would be non-specific. But of course, what you have to remember is, yes, they bind ATP in the active site. And the ATP binding component is going to be similar in all of those enzymes. But as you get a little bit outside of the active site, the sequence changes completely. So the ligands that bind these kinases recognize components that are different. They recognize different components that would be different in different kinases and provide specificity. So they turned out to be remarkable drugs. So Gleevec, or amitinib, was the first small molecule kinase that was approved for medical use. So it binds in the cleft, but it doesn't bind in the same way that ATP binds. And as you can see here, this is ATP, this is Gleevec binding in the active site. So it's recognizing components of the enzyme that are different. And this is an inhibitor of ABL kinase, and it's associated with a genetic rearrangement that leads to an aberrant protein that is constitutively active. It gives almost 100% remission if used in the early stages of the disease, leukemia. And so it's highly successful, and it's one of a very large number of kinases. So when you're watching television, if they have IB associated with whatever name is associated that is being sold or being advertised, it's a kinase inhibitor. So look carefully when you're watching adverts, and you will see how many of these are now on the market. So the summary of ATP binding motifs. And again, this is the big picture, and this is really important. They all share the use of metal ions to neutralize the negative charge. So in the case of the P loop, the negative charge is stabilized by metal ions. But the terminal lysine of the P loop motif is involved in neutralizing the charge on the gamma and beta phosphate. So they all contain components that neutralize the 4 negative charges that are present on the triphosphate moiety. And there may be one or more metal ions depending upon the class. There are no fixed rules about the side chains or main chain atoms to coordinate oxygens, excepting that they tend to be glycine rich, and it's to allow adjacent amide hydrogens to interact with phosphate oxygens. That would not be available for any other amino acid except for glycine. No fixed rules about the secondary structural elements, except that they tend to be strands rather than helices, which makes sense if you want to coordinate a extended molecule against a series of amino acid side chains. That is going to be better than an alpha helix. The positive side is that the common motifs are used ubiquitously to accomplish phosphoryl transfer. And the chemistry is similar in all cases. So once you understand one of these enzymes, or a couple of these enzymes, you can apply that understanding to all ATP-dependent enzymes. And finally, there are references, and there are a few references spread throughout the slides. If you have any questions about this lecture, please contact me. This is a wonderful subject, and as I said, it's a great opportunity to learn something about 9 to 10% of all proteins in our genomes. Thank you.